Bioinfocongress III. Dr. Joseph H. Davis, moleküler makine yapılarının derin öğrenme ve Cryo-Electron mikroskopi ile sorgulanması. Dr. Joseph H. Davis, 2003 yılında Berkeley'deki Kaliforniya Üniversitesi'nden hem bilgisayar bilimleri alanında hem de biyoloji mühendisliği alanında lisans derecelerini almıştır. 2010 yılında Massachusetts Teknoloji Enstitüsü'nde Profesör Doktor Tanya Baker ve Profesör Doktor Robert Sauer başkanlığında biyoloji doktorasını tamamlamıştır. Daha sonra Dr. Davis, Ginkgo Bioworks'te kıdemli bilim insanı olarak görev almıştır. Ayrıca Stanford Durham Prebys Medikal Keşif Enstitüsü'nde misafir bilim insanı ve Scripps Araştırma Enstitüsü'nde Jane Coffin Childs ve K99 üyesi olarak Dr. Jamie Williamson ile birlikte çalışmıştır. 2017 yılında Dr. Davis, biyoloji bölümünde Whitehead yardımcı doçenti olarak MIT'ye dönmüştür. Dr. Davis, Cryo-Electron mikroskopuna özel ilgi gösteren moleküler biyofizik ve yapısal biyoloji alanında araştırmalar yapmaktadır. Dr. Davis şu anda hücrelerin büyük moleküler makineleri nasıl hızlı ve verimli bir şekilde bir araya getirdiğini, düzenlediğini ve parçaladığını tespit etmek amacıyla çalışmalarını sürdürmektedir.
Bioinfocongress III. Assistant Professor Joseph Davis. Interrogating the structures of molecular machines through deep learning and Cryo-Electron microscopy. Dr. Joseph Davis received a Bachelor of Arts in Computer Science and a Bachelor of Science in Biological Engineering at the University of California, Berkeley in 2003. In 2010, he completed a PhD in biology at Massachusetts Institute of Technology under the direction of Professor Dr. Tanya Baker and Professor Dr. Robert Sauer. Afterwards, Dr. Davis started to work as a senior scientist of Ginkgo Bioworks and was subsequently a visiting scientist at the Stanford Durham Prebys Medical Discovery Institute and the Jane Coffin Childs and K99 Fellow at the Scripps Research Institute, working with Dr. Jamie Williamson. In 2017, Dr. Davis returned to MIT as the Whitehead Assistant Professor in the Department of Biology. His main research areas are molecular biophysics and structural biology, with a specific interest in Cryo-Electron microscopy. Dr. Davis is currently working to detect how cells assemble, regulate, and degrade massive molecular machines rapidly and efficiently.
Dear Dr. Joseph Davis and our participants, welcome all of you to our Bioinfocongress III event. Dr. Davis will give us a talk today on interrogating structures of molecular machines through deep learning and cryo-electron microscopy. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dear Dr. Davis, welcome back. If you wish, you can start your presentation.
Wonderful. Thanks so much for the introduction. Can folks see slides and hear me okay?
Yes, we can see your presentation. Everything is
perfect.
Great.
All right. Yeah, this has been wonderful. It's been really a couple nice talks I've been able to see, and thank you guys again for the invitation.
You're welcome.
So today I'm going to talk to you guys about some of my lab's work looking at macromolecular machines, specifically using an imaging technique that I'll describe, cryo-electron microscopy, and then how we've been trying to analyze that data using some techniques from machine learning. But I thought I would sort of start by just framing the types of questions that we're interested in answering. So, for a long time, we've known that these massive macromolecular machines, things like the ribosome or the proteasome, are absolutely critical for cells to live, right? And these are composed of mixtures of RNA and protein and other biopolymers. And they're really massive in size. So just as an example, the ribosome has about 50 proteins, 3 RNAs. Each of those RNAs have about 1,000 to 2,000 nucleic acids. And so there's this really large complex that needs to get built. And historically, we've been able to understand how these work by looking at their structures. Kevin sort of mentioned earlier today that, sequence we think sort of derives structure, and then structure gives rise to function. And these structures that we've been able to determine experimentally have really helped us understand how these machines work. Unfortunately, if you look at the terminal assemblies, you don't have a great sense of how they're built, right? So what do I mean by that? I'm basically asking the question of, how do you take a pile of individual components, so say all of the things that go into making a ribosome, and how do you properly fold and assemble them such that they give rise to a functional machine? And then if that machine becomes damaged or is no longer necessary, how might you disassemble it back to the constituent parts such that they could be recycled on some time scale? Okay, so that's the kind of questions that we're interested in asking. And one might ask, why hasn't this been a solvable problem previously? And I think really right now is an exciting time to look at this, in part because there's been 2 sort of technical advances made in the last 15 years or so. So the first are advances in quantitative mass spectrometry that really allow us to define the list of parts that go into each of these machines. So we can use mass spectrometry to sort of define all of the proteins that are necessary. And then very recently, advances in cryo-electron microscopy have allowed us to start to look at the structures of the assembly intermediates. And this has historically been difficult because these assembly intermediates are very heterogeneous. They're mixtures of partially assembled states, and those typically are not prone to, are not amenable to structural analysis because they don't crystallize, and they're just not very good for the traditional approaches that we use. However, cryo-EM has led us to start to look at that. Okay, so it's an exciting time to do this. And why might we want to? For a long time, we've known that the cell's ability to both assemble and disassemble these types of machines is strongly impacted by exposures to environmental stress, disease predisposing mutations, and those sorts of things. And so there's a large number of diseases that are associated with errors in the assembly or disassembly pathways. Okay, so today I'm going to tell you a couple stories really focused on the ribosome. And so I wanted to give you just a brief introduction to that system and why we think it's a useful system to think about assembly of these large complexes generally. Okay, so ribosomes, at least in bacteria, are transcribed from a primary RNA operon. So this is a depiction of RNA polymerase sort of moving along the primary operon. It first transcribes the small subunit RNA. This is known as the 16S. It then passes a tRNA and begins transcribing the 23S, so that's the large subunit RNA. And there's going to be an RNase that will come and cleave this tRNA out of the system and then liberate the large subunit RNA and the small subunit RNA, and those are going to go off and fold by their own folding pathways. They're going to undergo RNA modification and processing, and then a large number of ribosomal proteins are going to fold and bind to them. And so, in words, what has to happen? Well, you need to transcribe about 4,500 nucleotides. Those have to get cleaved into 3 separate RNAs, and those RNAs need to fold into about 150 helices. Concomitantly, you need to transcribe, translate, fold, and modify about 50 ribosomal proteins. And if you just think about the number of combinatorial ways that you could mix these, say, 53 components, it's this like preposterously large energy landscape, right? There's something on the order of 10 to the 70th potential assembly pathways if you just thought about the sequential ones. And yet, this process happens extremely rapidly and efficiently in all organisms. As an example, in E. coli, they make about 100,000 ribosomes per hour, that with each ribosome taking about 2 minutes, right? So this huge assembly has to come together, and somehow selection, evolution through selection, has found solutions to this assembly problem to do this incredibly rapidly and efficiently. Okay, so what do you get in the end? This is sort of an overview of what a ribosome looks like. On the left, this is the small subunit. You'll hear me refer to it as the 30S. On the right is the large subunit known as the 50S. And actually right between the two of them is where the message RNA is going to come, be decoded by the small subunit, and direct the elongation of the polypeptide chain by the large subunit. And those are actually done by these tRNAs that are shown in green here. What I want you to take away from this figure is sort of just to notice that there is a mixture of pink and red sort of throughout the structure, right? So the pink here is the ribosomal proteins. The ribosomal RNA is in this richer dark red color. And what you'll notice is that they're sort of nicely interwoven and highly intercalated, right? And so this is an interesting feature of the ribosome. It also creates a bit of a challenge in terms of assembly, right? If you were to think about making some RNA protein mixture, you might think about something like a virus, right? And in many cases there, you have sort of a core of nucleic acid and then a shell of protein around it. And if you thought about the assembly there, it's, you know, one sort of naive way you might think that assembles is that you sort of have this core of nucleic acid and then you have proteins that have RNA binding domains on them, and they can sort of use the core of nucleic acid to template themselves around it. That might be one potential assembly pathway. But that sort of naive approach isn't going to work here, right? You need to have these sort of intimate connections between the RNA and the protein that are sort of defined by these atomic contacts, and those need to be installed in the proper order. Okay, so how do we sort of think about models for how this thing might get built? You know, we might ask questions like, is there a single assembly pathway? That is, do you always put in component A, then B, then C, then D, or are there actually a whole bunch of parallel pathways, like the idea of like this factory being replicated multiple times? In some cases, you put in component 1 first, and in other cases, you put in component 4 first. You might ask, you know, are the components assembled individually? That is, do you put in every single screw one piece at a time, or do you sort of assemble modules and then drop those modules like an engine block into the car as it's being built? What happens if an assembly step gets blocked? So these cells are, you know, likely experiencing transient shortages in ribosomal proteins, or the availability of workers, when we call these like chaperones, or assembly cofactors. If there's transient shortages in those workers, what happens to the assembly line? Does the whole thing shut down and wait for that shortage to be resolved? Or does it figure out sort of parallel ways to continue building while it waits on that worker to come back? And then finally, if you make errors, how do those get corrected? Do you sort of fix the error in situ and then continue with assembly? Or do you recognize that there's an error, take that car off the assembly line and completely disassemble it and then put it back together? Okay, so what are some of the ways that we sort of look at this? We use a combination of quantitative mass spectrometry and cryo-EM. Today, I'm going to focus on our work using cryo-EM, and so I just wanted to quickly introduce that technique. So the way that this works is that you start with a grid, and just for general length scales, this is on the order of like 3 millimeters. You're going to take a couple microliters of sample. So this would be, in our case, sort of purified ribosomal assembly intermediates, suspended in some buffer. I'm going to apply a few microliters of that to the grid. And then this grid is going to get put in an electron microscope. And there's this very nice sort of movie that was at some point on Twitter, posted by Victor Chang and Karis Lau, that sort of shows first zooming in the visual spectrum and then going into electron microscopy. And so you can actually see the sort of data that we get. So if this will, oops, if this will play. There we go. So we're going to zoom in. I'm going to pause this at a couple sections. So this is what we would, you're starting to go into the EM range now. So what you can see here, there's this thin film of carbon, and cut into that film of carbon are a whole bunch of little holes. And we're going to zoom in further and further into those holes, and suspended in those holes is a very thin layer of vitreous ice, and it's within that vitreous ice where the molecules that we want to image actually live. So we're going to zoom in further and further and further here. We can now start to see this nice hole, and again, there's this thin layer of vitreous ice. And as we get closer and closer, you can now see these are individual protein complexes imaged with this technique. And so what's amazing about this is that you're getting, you know, tens of thousands to millions of pictures of individual molecules, all sort of suspended in this vitreous ice in random orientations and potentially in different conformations. And the challenge here is to try to use computational techniques to take these 2D projection images and reconstruct 3D density maps that allow us to interpret how the machine is assembled or how the machine functions in terms of the atomic contacts. Okay, now it plays. Great. So this is the challenge of single particle cryo-EM. You take, you know, these tens of thousands to millions of images. We're going to computationally align and average them. And when that works well, you can sort of see at the core of the molecule here, you can actually see the emergence of high resolution features from these relatively low resolution single particle images. However, when you align on that core, if the distal elements are actually in different conformations, they're not going to align well. And what you see is that there is some blurring sort of around the periphery here. Okay, so there's a couple ways that one can think about handling that. And we're going to talk about some of those ways. When you don't have the blurring, what is the goal here? So the goal is to use approaches like expectation maximization through iterative refinement algorithms to infer a 3D density map that best explains the underlying data. And so to do that, we need to both infer the density map and the angle with which each one of these 2D images were projected. So if everything lines up well, this is actually a relatively straightforward problem, and tools exist now that do this very, very, very well. What happens if there is, however, compositional or conformational heterogeneity? Right, so one naive approach is you could say, well, everything's not going to line up well. So what I'm going to do is I'm just going to pretend that there are two states. And so instead of trying to infer one single voxel array, I'm going to try to infer two voxel arrays. So I have to sort of assign particles to each one of those voxel arrays, still assign their poses, and then I can infer two 3D density maps. And you can sort of, naturally extend this to whatever sort of number of K you're able to computationally handle. But I would posit that there are sort of fundamental elements of these biological molecules that are just not well aligned with this discrete classification approach. And so as an example of that, imagine that we have some protein complex that can live in at least two states. One, where there's sort of a fixed region of the molecule, here this S shape, and there's a distal domain that can either be in the up position, or this thing can rotate down into state 2 in the bottom position. So we could imagine parameterizing that along a reaction coordinate. You might have state 1 in the up position and state 2 in the down position. And if this were an incredibly cooperative transition, I should really have this pointer back on. If this were an incredibly cooperative transition, then you might imagine that there's really just sort of two states that are populated. And this would actually be well handled by a discrete K equals 2 multi-class refinement, right? It would basically pick out this as state 1, all the particles corresponding to it, all of these particles as corresponding to state 2, and you could likely solve two relatively high resolution structures. Now, as I've defined this is a continuous transition. So even if it's cooperative, we know that at some level, there must be lowly populated intermediates that correspond to this sort of like intermediate states as this domain moves down through this trajectory. But if those are so low abundance that we can't really see them, we may not be able to study them, and they may not be of biological interest. The concern, however, happens as these become more and more populated. How do we handle them? Right, so there's sort of two things you can do. One is that you can just simply ignore them. And that would be to say, well, there's still relatively low abundance. I'm going to focus on state 1 and state 2, because with those states, I can get high resolution structures. But now you're throwing away, a significant portion of your data. The alternative might be to say, okay, well, I'm going to try to group them into one of my two states. And if you do that, I'll just tell you, you end up blurring one of the two states. And without having the molecular details, it's very hard to generate hypotheses that are testable. Okay, the most extreme version of this, of course, is that it's a completely continuous transition. All intermediates are populated, and now there's not really a good way to handle it. You can either sort of blur both of your states, or you can throw away the vast majority of your data and pretend that this is a highly cooperative transition. Okay, now the biggest danger about this is that you can sort of infer molecular mechanisms based on ideas of cooperativity that could be completely wrong. And if you go and look at the literature in typical cryo-EM experiments, the vast majority of the data is thrown away. Everyone does this. I'm guilty of this too. When I was getting started in my postdoc, we had this sort of interesting paper about ribosome assembly. And one of the big conclusions was that it's a highly parallel, but also highly cooperative process. And the reason we came to that conclusion is that we did the most extensive classifications that we could, given the data that we had in I did this work in 2014, 2015. And we couldn't find intermediate states between some of our key structural states. So just as an example, if you look at this assembly intermediate, sort of in density maps, in red here is what we could resolve, and in gray is what you would expect from a fully mature ribosome. And we couldn't find any intermediates between this red density map and this green density map. And what that would imply is that there is a highly cooperative assembly going from this state to this state. And if you go and look at an atomic model, that would include the assembly of 5 proteins, and about 14 rRNA helices would need to get folded and docked into their native position. And that certainly could be what's happening. The cell could be doing this extremely cooperatively, and maybe that's what helps to drive the assembly towards the terminal state. My big concern was, or this is a sort of an artifact of the way that we use discrete classification in these imaging data sets, and we've just lost a bunch of these intermediates. And so really when I started my group here at MIT, that led me to ask this question of like, how might we better model continuous structural trajectories? And that's what I'm going to talk to you about today. Okay, so the big idea here is to start to think about a functional definition of structure. And what do I mean by that? So what I've shown you previously is that you try to simultaneously infer pose angles for your particles and a 3D voxel array that is consistent with those particles and those poses. Another way to think about structure is to say, I just want to imagine that I have a function where if I give it some position in Cartesian coordinates, some x, y, and z position, it tells me the electron microscopy density at that position. Okay. So if I wanted to generate a 3D density map using a function like this, I would basically just rasterize along these x, y, and z coordinates at each point getting the density, and what would come out of that would be some density map. So if you just did this as I've described, it's not really that different than directly inferring the 3D lattice. You still get a single structure out of it. But if we start to frame this idea as a functional definition of structure, then you can imagine a function that's a little bit more complicated, whereas instead of just taking the x, y, and z position, it takes in some additional variable, say a discrete class k. And so now conditioned on which discrete class k you give it, you get back different density maps as you rasterize through. And if you can imagine this, well, then you can imagine a potentially more complicated function that takes a continuous variable z, and condition on that continuous variable z gives you different density maps. Right, and so this sort of pie in the sky dream of this is that instead of a single reaction coordinate, you could give this this sort of black box some location in a multi-dimensional energy landscape, and you would get back a density map based on the position in that energy landscape. Okay, so how could one learn such a complicated function? Well, one, you'd need data that gave you sort of some sense of the structure and the populations of those structures so that you could infer this energy landscape. And second, you'd need to learn a way to sort of use that data to train or to learn this black box function. And so the idea was that, you know, potentially MLPs could be the function that could map from these multidimensional energy landscapes to 3D density maps. And so really this was sort of the work of an incredibly talented graduate student, actually the first graduate student to join my lab, Ellen Zhang, and Tristan Bepler, who was a graduate student in Bonnie Berger's group. And the way that they tried to imagine this was more or less what I've described here. So they said, okay, well, you could take some 3D position, some coordinate position in this 3D voxel array. They're going to use a positional encoder and then pass that to a neural network. They're going to train this neural network by having it infer the density at that position. That gives us a model slice. And then they compare that model slice to one of the input images. This is our real data, and use the difference between these as a loss function to update this, this neural network weights. And so we're going to train a new neural network for each one of our data sets, basically passing through it a whole bunch of our images until it is stabilized and learned to map from these 3D Cartesian coordinates to this model slice. And then, of course, to get a density map, we just iterate across that whole thing. Okay, so this is a little bit different way to think about structure, and we call these, you know, they've sort of gained popularity in the ML field since we worked on this. They're called neural radiance fields. So the first question we tried to ask was, you know, can these neural networks learn 3D density maps from these relatively low resolution structures? And it turns out they can. So what I'm showing you here on the left is now a structure determined by our tool we call Cryo-Dragon, assuming that all the molecules are homogeneous. And you can see that this map looks very similar to the traditional approach. This is a tool called CryoSpark that uses traditional reconstruction. And these density maps are largely interchangeable. And just to get a sense of the types of features you can see, you know, this is a protein alpha helix sort of along this axis. And you can sort of see the pitch of the helix. And in some cases, you can actually see large bulky side chains. This gets quantified with something called a Fourier shell correlation. I'll sort of skip that for now. But the only thing that I want you to take away from it is that, as you'd expect for a neural network, the sort of representation capacity of the network, as you get bigger and bigger, allows you to achieve higher and higher resolutions. And at the higher resolution features, again, we can sort of see the molecular details that allow us to generate testable hypotheses. Okay, so this allows us to sort of look at static structures. Can we also simultaneously learn heterogeneity? Okay, so this is now structured as a variational autoencoder. The idea here is that you have an encoder network. So it's going to take each input image, sort of map that to some low dimensional latent space. And now our decoder, instead of just taking a positional code, is also going to take this latent code. So this is that sort of conditioned variable that I mentioned at the beginning of this section. And based on the concatenated latent code and the positional code, it's going to give us a density. And again, that density gets compared to the input image through some loss function. And that loss function then updates the weights and biases of both the encoder and decoder networks. Okay, so we're going to pass the whole data set through this epoch after epoch after epoch, and the encoder is going to learn to sort of separate out in latent space structurally different molecules, and the decoder is going to learn to generate 3D volumes from those particles. That's our hope. Okay, so we first tested this with simulated data, so we've actually just took the example that I showed you before. We're going to start with, excuse me, we're going to start with one conformation. We're going to rotate this domain down, generate a bunch of volumes. From those, we can then sample in different regimes. So we can sample it as a highly cooperative 3-state transition, or a perfectly cooperative 3-state transition, a highly cooperative one, or a completely continuous reaction coordinate. And then we can look at what the learned latent space looks like relative to these distributions. And you can see that there's a nice correspondence between the learned reaction coordinate and the input distribution. Okay. So how does this sort of work? If you look at the input distribution and the learned distribution, you can see it after 0 epochs of training, or when you start, you just assume everything is the same. But then over epochs of training, you can see that quickly the encoder learns to separate these things out. And one thing you should notice here is, this is not a perfect correspondence between the learned distribution and the input distribution. You see that most strongly sort of in the continuous, right? That the prior, which was a unit normal, is sort of left over in this learned latent distribution. But that being said, as we sample along it, we get back maps that map to the ground truth maps. Okay, so we can learn this heterogeneity by encoding images into this low dimensional latent space and then decoding them to get back volumes. All right, so how do we use this in practice? So again, we have this variational autoencoder we call Cryo-Dragon. Particle images get passed through it. They get mapped into a low dimensional latent space and then get decoded into a density map. After it's trained, you can pass your images through one more time, and now you can actually visualize this learned latent space. And we found this to be a rather useful exercise. The learned latent space tends to be organized by structure, and that allows us to then find related particles. So you can select those related particles and then validate the structures that you get back with traditional tools. We've also found it's a very effective particle filtering tool. Things that get sort of pushed out of distribution are often sort of artifacts of the imaging process, and those can be filtered out of your data set. You can sample from discrete locations throughout the learned latent space, and what we find is that you find these discrete states. And then, of course, because this is a continuous and generative model, you can actually walk a traversal through the learned latent space and get these continuous trajectories. Okay, so how do we like to use this? So we use this to find rare but biologically informative states. It allows us to quantify the relative abundance of these various structural states and potentially discover structural intermediates. And then most critically, this sort of we see as a hypothesis generating tool that allows us to relate some experimental conditions to structures. And then using those structures, we can think about how the sequence is allowing this assembly to occur, go in and make mutations there, and then see how that perturbs the assembly pathways. Okay, so I'm going to show you just sort of one vignette of how we've been using this. And so this is the work of another very, very talented student in my lab, Laurel Kinman, working in collaboration with Joaquin Ortega's group. And Jingyu Sun is a graduate student who also sort of helped lead this work. Okay, so the story I want to tell you about, again, involves ribosome assembly, and specifically here asking the questions of how a particular assembly factor, so this is like a chaperone that helps the assembly get built, how does that impact ribosome assembly? So for a long time, we've known that these chaperones are important, but determining exactly where and how they act has been different. And so what we're going to do is we're going to try to visualize the chaperone-bound particles and use these structures to infer how they act. The one that we're going to talk about is called KSGA. It turns out this is a non-essential RNA methyltransferase, but it's highly conserved. So you see homologs of it in all eukaryotes as well as prokaryotes. Okay, so KSGA binds to the 30S. There's actually a low resolution structure available there, and it chemically modifies the ribosomal RNA. But seeing where it bound and knowing what it modified didn't actually help us understand its role in assembly. And we know that it's an assembly factor because if you knock it out, you get an accumulation of a bunch of assembly intermediates, particularly under cold growth conditions. Okay, so what Jingyu and Joaquin's group did is he basically made genetic ablation of KSGA in E. coli cells. Those are cold sensitive. They grow relatively slowly, but from them you can isolate ribosomes. And then you can do one of two things with those. You can either directly image them by electron microscopy, or you can take in vitro purified KSGA, add them to these particles, and then plunge freeze that sample. And the idea is that we're going to then use Cryo-Dragon to compare the structural ensembles from treatment with KSGA to untreated cells. Okay, so the first data set I want to look at is upon treatment of KSGA, what do we see? So the first thing that we notice is that the latent space is highly featured. And in our experience, this often corresponds to enormous structural heterogeneity. And if we go and sample from positions in latent space, that is exactly what we see. So sort of in an outline here, this is what the mature ribosome would look like. And then in the dark colors is actually what we see in the density maps. And so you can see that there are some structures that are missing entire domains. Here, the head and the platform. There are others that are missing only the platform and not the head. There are still other structures that look almost entirely mature. And then finally, you see structures, as you'd expect, with KSGA bound. Okay. So just to sort of sanity check that this approach is sort of giving us the types of structures you might expect, we also use traditional tools with discrete classification to see what types of structures we can find. And you can see by traditional classification, although it's a bit onerous and requires a bunch of expert intervention, you can actually find all of these types of structures that give a nice one-to-one correspondence between what we find in Cryo-Dragon and what you see by traditional tools. Okay, I sort of motivated all this by saying, well, we don't like discrete classification because you don't know how many classes there should be. So, of course, the obvious thing to do is to try to use this generative model to better characterize structural heterogeneity. And I would say, just thinking about these generative models, they actually sort of create their own problems because they are so rich in their representation capacity that it's hard to then think about the structures that you get back and use those to generate hypotheses. And so we've been thinking about, how to use tools like Cryo-Dragon quite a bit recently. The approach that Laurel came up with was to say, okay, well, I can generate an arbitrary number of structures, so I'm going to go ahead and generate a ton of them. So in this case, she generated 500 structures, and she used k-means classification cluster centers just as a way to sort of nicely sample across all the space. The particles that go into each cluster turns out doesn't matter. We're just using this to sort of define locations throughout latent space to sample from. Okay, so now once she's defined these spaces, she can generate 3D density maps from all of those spaces. Great, that solves a problem. We now have density maps, but it creates a new problem of now we have 500 density maps, and how do we start to think about those? So what we thought is, well, we're sort of in a unique position in that we have atomic models for what the mature ribosome should look like. And so what we could do is simply ask, for every sort of component of the ribosome, be it a ribosomal protein or an RNA helix, is it there or is it not there? A very sort of straightforward, naive way to think about this problem. And you could do that by sort of calculating the fraction of the EM density that you observe relative to what you would expect to observe if that component were there. Okay, what do I mean by that, right? Let me just go through an example. So here I have two different density maps that Laurel sampled, one in orange and one in blue. In this green is the atomic model for one particular helix called helix 44. And so using this atomic model, we can simulate what the density is that we would expect to see in this location. We can calculate the expected density relative to what we observe. And that gives us some occupancy value that scales 0 to 1. And so, for example, for helix 44, in the orange map, it's fully occupied. It's completely unoccupied in the blue map. And in contrast, helix 15, which is this helix, is fully occupied in both of them. Okay, so this gives us a nice metric now to look at hundreds to thousands of 3D density maps and to look at all of the structural elements within them. And so we can just plot that as a clustered heat map. Okay, and so here's what sort of falls out of it. Each one of these columns is a different structural element, either a ribosomal protein or an RNA helix. And each one of the rows is one of the density maps that Laurel generated using Cryo-Dragon. And you can see that there are sort of major groupings, both of structural elements, sort of denoted here by these different colors that come out. And those are things that show similar patterns across all the density maps. And then there are groups of density maps that show similar patterns across the structural elements. Okay, so you could say, well, I'm going to define these structural blocks by just adding sort of a threshold on this dendrogram. And I could ask, where do they map onto the structure? And sort of satisfyingly, these actually organize in a way that makes sense. You can identify the body domain. This is a large folded domain that was known to fold cooperatively, and that is distinct from the head. And so the sort of baseline structural elements make sense. We could ask questions like, is the block interconversion, say going from an immature to a mature structure, obligately ordered? And the way you might ask that is to say, well, I have evidence of fully mature structures, and I have evidence of fully immature structures. Can I find evidence of structures that allow me to transition between these two? And of course you can. And what you find is that there are some density maps that allow you to build the head first, but they still lack the platform. There are other density maps that imply the platform gets built first, but not the head. And because these two have mutually exclusive occupancy, we propose that that implies parallel assembly or disassembly, so what I'll call interconversion, right? So you could either build in the platform and then the head, or you could build in the head and then the platform. And so that says that those two platform and head domains are sort of operating independently. We can ask questions like, what types of particles can KSGA bound? Previously, I showed you that there was a structure of KSGA bound, and we know that it can bind to the fully mature particle. We are curious, what other types of particles can it bind? Does it require that the particle is fully mature, or can it bind to assembly intermediates? And so we can basically use this occupancy map, define the sets of volumes that have KSGA well occupied, take that subset and recluster it. And what comes out of it is a cluster map that looks like this. And again, we can look at maps from them. And so we can find density maps where the particle looks very mature. It has the head, the platform, the body, and it's just missing this helix 44, which turns out to be at the same binding site of KSGA. We can find other density maps that are completely lacking the platform but have the head. And we can even find KSGA binds to particles that are lacking both the head and the platform. That are lacking just the head but not the platform. And so this really implies that the local determinants of the binding site for KSGA are what allows it to bind, and not really these global determinants, right? And so that's sort of an interesting finding, and it makes us think differently about sort of what this factor is doing. Okay, we're next going to look at the untreated control sample. And so the expectation here was, okay, we saw a bunch of immature assembly intermediates when we treat with KSGA. KSGA is an assembly factor, so our expectation is that you'll see even less mature material in this new data set. What was sort of surprising is that, this is why we do science, when you go and look at it, the density maps that you find are actually much more mature. It's very rare for us to find density maps that don't have the head. There was only one of them, one of the classes here in a relatively small class. And that was in contrast to the KSGA treated. So to us, this sort of implied that upon treatment with KSGA, you were actually sort of globally disassembling the ribosome. And so we started thinking about, well, why might the cell be doing that? Why might evolution have selected for KSGA to disassemble particles? And one thought was, well, perhaps these things sort of grossly look mature, right? They have the head, the platform, the body, and this helix 44, but maybe there's sort of subtle errors in them. And KSGA is recognizing these subtle errors, and the only way to fix them is actually to disassemble the particle. Okay, so one of the hints that this might have been true was that we can look at the occupancy for any particular helix. And we knew that helix 44 was sort of the critical linchpin helix to make ribosomes work. And so we looked at its occupancy either in KSGA treated samples or untreated samples. And the interesting thing was that in the KSGA treated samples, it's basically never present, and that sort of makes sense. You know, the binding site is very close to the helix. But in the untreated sample, we found this sort of multimodal distribution where the occupancy wasn't just like 0 or 1. It was a bunch of partial occupancy, and that's often in our hands been typical when things are undergoing conformational heterogeneity. Okay, so we wanted to sort of inspect helix 44 a bit more closely. The way that we did that is that we identified the particles from each data set that had helix 44 occupied, and then we generated volumes for those. We then produced a mask that just focused on the sort of region of helix 44, and we used principal component analysis on those density maps to try to find what are the dominant modes of motion within it. Okay. And so what we were really excited to see was that there were two major sort of endpoints of this dominant mode of motion. One where the helix shown in orange here is sort of tucked into the body of the ribosome, and one where it's released and sort of pushed out. And if you go back and look at the literature as early as 1969, Elson et al. had sort of predicted that there should be two conformations of helix 44, one which was active and one which was inactive. Okay. And so if you go and look at sort of a traversal along that principal component of motion, you can actually see this is sort of sets of experimentally determined structures we have where the helix pops out, and then I'm going to play it in reverse, it popping back in. Okay, so within our data sets, there was evidence that this helix can adopt either an active or inactive conformation. And so the next obvious question is, you know, what are the distributions of those two conformations, either in the KSGA treated or the KSGA untreated data sets? Okay, so that's an easy experiment to do. You can just look at the density along that principal component of either KSGA treated samples or untreated samples. And hopefully this image is like sufficiently clear, but in the untreated samples, you can see this bimodal distribution. And if you sample along it, you can see that that corresponds to both helix 44 active and then this sort of inactive where it's pulled away conformations. And what you'll see is when you treat with KSGA, it's this population of particles that are specifically pruned away. Right, so it's as though treatment with KSGA removes this population. That population, sort of through historical biochemistry, was known to be inactive. And so it actually looks like KSGA is proofreading these helix 44 inactive conformations. Okay. So what are the impacts of this proofreading? So we went and looked closely at our structures, and they were actually sufficiently high resolution that we could build atomic models for them. And the sort of interesting thing here is that there's what we call a linker helix that's formed that sort of links helix 44, helix 45 through this linker helix to the rest of the structure that actually connects the sort of body, platform, and the head domain. And what's sort of fascinating is that if you disrupt helix 44, that impacts this linker helix and then helix 28 that links to the head. And so we hypothesized that if you displace helix 44, you should actually sort of release the head and turn this thing into like a bobblehead, more or less. And if this were true, it might explain why we see sort of grossly less mature looking structures when you treat with KSGA. The idea is KSGA is recognizing the inactive helix 44. It comes, remodels that somehow, and in doing so, it sort of releases the head to float around. Okay. So we can use this same technique. We can count the number of particles that either have the head or don't have the head with KSGA treatment or without. And so as quantified here, you can see that upon KSGA treatment, you have a whole bunch more headless particles. We can put a mask around the head and then ask what happens to it. And so you can actually directly visualize some of these motions. And so you can see here in orange, this mask, you can see this head domain sort of bouncing around in different conformations. And again, we hypothesize that's resulting from KSGA binding and disrupting the network of RNAs that sort of hold the head in this place. Okay. We did some mass spec experiments to show that the head actually remains associated with the particle. It's not as though it's completely denatured, but we think it's just undocked and therefore flexible. Okay, so overall, what does this tell us about the activity of KSGA? So, go ahead. So the basic idea would be that when KSGA is sort of depleted from cells, or if there are transient shortages of it, with some frequency, particles assemble into an inactive form. Treatment with KSGA recognizes this inactive form. It disrupts helix 44, and in doing so, it actually sort of proofreads the particle, resulting in a less mature particle where helix 44 is displaced and now the head is undocked. So that seems like a bad thing, but what could then occur is that now you get another chance to assemble, and through this other chance to assemble, you have a chance to form the 30s particle with an active helix 44. And I should just say, okay, so this is the sort of second chance. Of course, particles can assemble directly in the absence of KSGA. It's not an essential factor, and so this pathway must exist. The last thing I should say about this model is that, you know, we only have a single sort of static snapshot, and so we can't actually distinguish whether parallel assembly is occurring, and that's what we observed with this sort of mature, immature, platform first, head first, or whether it's parallel disassembly, that KSGA binding gives rise to a bunch of different ways to disassemble the particle, proofreading activity still occurs, and then you mature. Okay, so this is sort of one of the ways that we're using CryoDragon. And so where does this leave us? You know, I think that this gives us the opportunity to find rare but biologically informative states. I sort of showed you how we can find that KSGA binds to a huge variety of assembly intermediates as opposed to simply the fully mature particle that had been reported previously. It allows us to sort of quantify the relative changes and distributions of elements within these large complexes. And so in general, you can use this to quantify the abundance of structural states. It allows us to find new structural interdependencies. I realized I didn't actually show you this portion of the data, but I'm happy to talk about it at the end if people are interested. And then finally, it allows us to test hypotheses that relate the experimental condition to structure, right? So we could see the impact of KSGA binding to small subunit structural intermediates. Okay, so where are we going with this? The newest things that we're working on actually allow us to now look at these heterogeneous structures directly in the cellular context. So what I've shown you so far is that we make some genetic mutations to the cell. We biochemically isolate material from them. Those purifications, take between hours and days, and then we image that material and we sort of say, well, we did our best to purify it in native conditions, and therefore we think that this reflects what was happening in the cell. But of course, all this time has elapsed. It's been exposed to all sorts of buffers. It's lost all of its native contacts with the rest of the cellular components, and so it's a little bit hard to really make that argument. And so what we'd love to do is directly look at structures in the cell, and there's sort of an emerging technique called cryo-electron tomography related to cryo-electron microscopy that might allow us to do this. So the way that this works is that you take cells, you actually grow them on these same cryo-EM grids. They're too thick to image directly, but you can cut them with something called a fib mill into a thin lamella that is sufficiently thin that you can image through. And then you can, instead of collecting one single image, you can collect a tilt series. So you sort of tilt the grid side to side, and that allows you to get a bunch of different angles of the same molecule. The downside is that you have even lower signal to noise than what I had shown you previously. Okay. And so this is what a sort of tilt series looks like. This is from a paper from Yulia Mohammed's group and Dmitry Turganov, really one of the more beautiful examples of cryo-electron tomography. This is a bacterial cell, and the little dark dots, well, there's two types of dark dots. One of the very dark ones you should ignore. The other ones, these sort of faint dark dots, those are actually ribosomes in this bacterial cell that was grown and then frozen on a grid. Okay, so Barrett Powell, yet another talented graduate student in my lab, took it as his challenge to figure out whether he could adapt this CryoDragon framework to work with this cryo-electron tomography data sets. Okay, so what does this look like? Right, again, you take this unaligned tilt series. From this tilt series, you can reconstruct a tomogram. So this is a 3D voxel array of the entire cell. It sort of looks like this. And then from this reconstructed tomogram, you can pick out individual ribosomal particles, and there are nice algorithms to do this. And you can take all these individual picked particles, align them to one another in the same way that you do with traditional reconstructions. And this is still just mind-boggling to me, but doing that, you can then reconstruct ribosomes. Oops, something is jumping ahead here. You can actually reconstruct structures of ribosomes as imaged in the cell. And Dimitri's work was just really sort of like transformative for me personally, I think for the whole field, when he was able to resolve density for small molecule chloramphenicol in a cell, right? This is an antibiotic they treated the cells with. He can actually find density for it in these maps. And so it's as though you're actually imaging a drug on its target in the cell. I think it's just absolutely incredible. Okay, so this is sort of akin to traditional cryo-EM reconstruction. You get back a single static structure, even though we know that these things are dynamic and moving around and sampling all sorts of conformations. And so the idea was, can we convert from this beautiful but static structure to something more dynamic using CryoDragon? And so I won't go through the details in the interest of time of how Barrett's done this, but he's basically adapted the encoder network to now handle these sets of images that are all related by some tilt angle. And we have to change some of the loss function, some of the other details here to be able to do these reconstructions. But the idea, again, is that you should embed now subtomogram averages, or sorry, subtomograms into a low-dimensional latent space, and from that low-dimensional latent space, be able to decode 3D volumes. Okay, so does this type of approach work with this new data type, which again is even lower signal to noise than cryo-EM data? And so Barrett showed that he too can reconstruct density maps of the ribosome at resolutions that are sort of useful for molecular biologists thinking about, atomic contacts, or at least like domain contacts. And this scales sort of as you would expect. With longer training, you go to higher resolution. It's shown in the blue to red here. And with a larger network and higher dimension, higher resolution images, you also go to higher resolution. On the encoder side, we next asked, again, does this encoder network encode these particles into a latent space in a representation that is useful? So he took Dimitri's data set, ran this encoder network, and the first thing that we noticed was that there were two major groupings here. And this was kind of interesting. If we looked at the grouping on the left here, what we immediately noticed was that these didn't actually look at all like ribosomes. And if we went back and looked at the particles that gave rise to this group, they were often particle picks sort of even outside of the cell. You can see some of these in green here. And so again, this was evidence that this CryoDragon framework is a really effective particle filter. So there are some false positives that come out of the picking algorithms, and CryoDragon is able to filter them away. Once we've removed them from our data set, we can train a new CryoDragon model, or TomoDragon model. And with that trained TomoDragon model, we can again sample 3D density maps throughout latent space. And again, this was a pretty exciting result. If we, for example, looked at volumes in this region, not only do we see a 70S ribosome, but we can actually find evidence of a lipid bilayer membrane with some density beyond it. And if you go and look at the particles that are in this region of latent space, they all sit along the periphery of the cell. And so what we think is sort of, more or less accidentally, because we have this unbiased model to look at heterogeneity, we found sort of ribosomes that are actively translating or going through co-translational translocation, secreting material out into the extracellular region. Because they happen to be ribosomes, we were able to find them. This density on the outside of it is very low resolution, but it actually fits quite nicely with an atomic model of the SEC-DF translocon. So we think that this, again, is an actively translocating ribosome, sending its peptide product across the lipid membrane into the extracellular space. So what's really exciting about this is that we can get all these different types of structures, and now we can map them back into the cellular context. And that's sort of what's shown here. Barrett's just sort of categorized these in different structural states of the ribosome. And then you can see their spatial distribution, again, within this bacterial cell. Okay, so I think I'll stop there, and I would love to take some questions, but I do want to just really quickly acknowledge the folks that did this work. So Ellen Zhang, shown here, recently graduated from my lab and has started her own research group at Princeton in the computer science department. I'm incredibly proud of all the work that she's done. Fantastic scientist. Laurel Kinman is starting her 5th year graduate student in my lab who did all the work with computational analyses of KSGA. And then Barrett Powell is also a 5th year graduate student who did all of the tomographic work that I described at the very end of the talk. And then again, I'd like to thank Joaquin Ortega and Jingyu Sun from McGill University, who collaborated with us on the KSGA project and really helped us think about and understand those structures. And then thank the funding agencies for supporting this work. Happy to take your questions. And
First of all, thank you very much for your very informative presentation. The first question is, does cryo-EM tomography is useful for mammalian cells and give information about protein-protein interactions?
Yeah, so actually, there's probably more applications for cryo-ET in mammalian cells. There's a couple different challenges there than we've had to handle. So there you typically grow the cells directly on the grid, but then you go through the same process of fib milling them to cut them down to be thin enough and then image them. Let's see, for us, we grow the cells sort of in liquid cultures and then concentrate them, put them on grids and freeze them. So subtle differences there. The other challenge is just the length scales in mammalian cells, right? They're enormous. The tomograms that we generate are relatively small by comparison, especially at these higher resolutions. And so you only see like a small window of the mammalian cell with any particular tomogram. And so the challenge there is if you're looking for rare events, like let's say that you're interested in, I don't know, maybe it's like a contact site, say, between two organelles, maybe the ER and, I don't know, say a lysosome or something like that. Those events could be rare, and it's unlikely that if you just randomly look at a small window in the cell, you're going to see it. And so there's a related technique called CLIM, which is correlated light and electron microscopy, where you can use sort of genetically encoded fluorescent tags to help you localize within the cell the region that has the feature that you're looking for. And then you can use that as a signal to go in and cut your window just in that region and then go image it. But I didn't talk about any of that work today.
Thank you very much for your answer. If you wish, you can close your presentation and you can see us too.
Yep.
So thank you.
Did that work? Yeah.
Yes.
Okay.
I move on with the next question. Why the ribosome is targets for such studies?
Yeah, so for the assembly studies, I think it's just a super interesting problem because it has to co-fold RNAs and proteins. And the RNAs are really subject to a lot of kinetic traps. You know, there is sort of one conformation of those RNAs that gives rise to an active ribosome. But there are a ton of sort of related structures that are similar in energetics. They're stable, but they're not active. And so somehow evolution has selected for assembly pathways that help the RNA find this active conformation. We think that that's part of what these chaperones do. It's also likely part of what the ribosomal proteins are there to do, is that you get small gains in RNA folding. Maybe you form the proper helix, or that helix properly docks with another helix, and that creates a binding site for a ribosomal protein to come in and sort of lock in those gains in RNA folding. Whereas if you didn't have the proteins or you didn't have the chaperones, you might transiently fold that, but the likelihood of getting the hundreds to thousands of those transient interactions all at the same time is very, very low. And so we think this creates like a really interesting assembly challenge to try to study.
Yeah, I understand. Thank you for your answer. I move on with next question from YouTube. What limitation exists with neural network-based reconstruction methods?
Yeah, I think that there are sort of there's at least two big limitations. I think one of them is that at least in our hands, we can get to resolutions that are useful, but we're still nowhere near the sort of state-of-the-art resolutions that you can achieve with traditional approaches. So, the best reconstructions we can get with neural networks are sort of in the, let's just say, 3 angstrom regime. And the sort of state-of-the-art resolutions one can achieve with traditional approaches are, I don't know, just call them like 2 angstroms, not to create controversy. So it doesn't sound like a big difference, but it turns out the sort of detailed atomic resolution things that you can see at 3 angstroms versus 2 angstroms is pretty different. And so we're not able to achieve that. I think the other challenge is that there is, there is still sort of a fear of neural network hallucination of the structures that you see. With traditional approaches, we have sort of nicely defined ways to do, data set splitting and validation and cross-validation, so that we know the structures we get out are not these hallucinations. And there are ways we could do these with these neural networks, but we haven't actually done that yet. And so what we often have to do is take our neural network reconstruction, choose the particles that are most close to it, and do a traditional reconstruction with it to convince ourselves that that's not a hallucination of the network, but is in fact something in the underlying data.
Thank you for your answer. I continue with next question. How Tommy Dragon recognize membrane proteins?
Yeah, so yeah, like why did it all of a sudden find all these membrane proteins and then sort them out in latent space all on its own? This is like the magic of deep learning, right? So at some level, what it's basically telling you is that the input images, the images that were taken of these ribosomes associated with the membrane, they have some feature that makes them very, very distinct from all the other ribosomes. And of course, that feature is the presence, is the presence of the lipid bilayer, right? So there is a whole bunch of pixels at the bottom of the ribosome that are only there for the membrane-associated ribosomes and aren't there for all the cytosolic ribosomes. And so because that feature is such a strong feature in it, the encoder network learns to separate those out such that the decoder network can reconstruct the membrane from this set of particles, but not from this set of particles. But what's really cool about it is that this is a completely unsupervised discovery of this separation of particles.
Thank you very much. Next question from YouTube. Does Cryo-Dragon applicable to multiple particles? And if it is, how might that collaborate?
So I'm not sure I totally understand the question, but my my rough understanding is that like what I've shown you is if you have a whole bunch of related particles like ribosomes that are in different structural states, we've shown that Cryo-Dragon sort of nicely separates these subtly different structural states. And maybe the question is, if I had ribosomes and proteasomes, what would Cryo-Dragon do with that? So one of the things that the Cryo-Dragon implementation I described for you so far requires is that you have sort of the poses or the angles with which you image the molecule already assigned. And we get those angles by performing what we call a consensus traditional reconstruction, which is basically just taking all the ribosomes as best we can, aligning them to each other so that they're all in the same reference frame. And that works well if you have like a large domain that's easy to align on, and then the other domains, even if they're moving around, they don't affect the overall sort of assignment of pose very much. But if you're going to try to mix ribosomes and proteasomes, that's not going to work because it's hard to get, it just doesn't make sense to put those in the same reference frame. They're fundamentally different molecules. So I think it's an interesting idea. Like one of the things that we've been working on, and there's a preprint that that is posted now, is to, in the Cryo-Dragon framework, do on-the-pose assignment, on-the-fly assignment of pose. And so that might allow us to get into even more heterogeneity where there are mixtures of particles that don't align well to each other. But those methods right now are very, very slow and still sort of experimental.
I understand. Thank you for your answer. Next question is, which optimizations are required for the ability of Cryo-Dragon to provide a low-dimensional representation of the heterogeneity of the data set given by a latent coding of each particle?
Yeah, so my guess here is that you're thinking about sort of what are the hyperparameters that need to go into the model training, and like to what extent do those hyperparameters impact the ability of the work for this to work? We did, like my lab, basically, we put this together, this idea together right around the time when COVID hit. So like it was sort of late 2019, early 2020. And so when COVID hit and my whole experimental lab was shut down, we basically had the whole lab use Cryo-Dragon and try it on a ton of different data sets that all had like different sorts of features. And that really let us narrow in on a set of good hyperparameters and heuristics to use for most data sets. And so in general, the sort of network architectures that we provide as defaults should work quite well for you. That being said, we have found examples where, you need to change, for example, the size of the encoder or decoder networks to really sort of have the representation capacity required for the heterogeneity that you're trying to encode. And so it is a parameter you can play with. Same thing with like learning rate and weight decay, or batch size for that matter. Yeah.
Thank you for your answer. I continue with a question from YouTube. A variational autoencoder has a reputation for producing blurred images.
Yep.
Thus, based on this approach, the reconstruction image may not be optimal with respect to the resolution, which might be critical for cryo-EM reconstruction. Therefore, what do you think in this situation? Thank you.
Yeah, this is a great question. This is the same feedback that we got from all the ML community when we first started thinking about using a variational autoencoder, was that it's going to be too low resolution. I think, in our hands, we're seeing the sort of resolution that we think is useful for the types of questions we want to answer. That being said, are there other architectures that that might produce higher resolution? Potentially. We've tried using GANs. We've tried like a couple other things. In our hands, those have been very, very unstable to train. And so we think that like the trade-off there has not been worth it. But this is an area of active exploration. And I think that there's, it's important for people to try a bunch of different approaches. There's like a related approach from Steve Ludge's group called E2GMM, another generative model that uses a Gaussian mixture model that I think has some nice ideas in it. There are some other folks that have gotten GANs to train more stably in their hands. And so, yeah, I think it's an interesting area. Again, for us, at least, like the size of particles that we work in and the sort of like major domain scale rearrangements that we're interested in seeing, the resolutions we get out of CryoDragon as a VAE seem to be sufficient, but that may not be true for everyone's questions.
Thank you for your answer. Next question is, does CryoDragon fully extract structural information from noisy images with the extensive processing power of neural networks?
Yeah, it's a good question. Like, I guess the question is like, is there additional signal left on the table that we're not?
Yes, from noisy images.
Yeah, yeah, it's an interesting question. I mean, I think that to some extent that might be true, right? Like we sort of showed that we don't actually completely reach the resolution that one achieves with traditional approaches. So why am I hesitating to answer this question? One is that it's a little bit hard to quantify exactly how much information is in these quote unquote noisy images. And that's for two reasons. One is, okay, of course you can simulate noisy images and then you know exactly how much signal is there. Those simulations are hard to do because it's very difficult to properly simulate all of the imaging artifacts that come out of a cryo-EM experiment. And so if you look at simulated, quote unquote, simulated noisy data, it just doesn't look like real particles to anyone that actually looks at these things a bunch. So there's features there that are just hard to simulate. The other way you can do it is to take real data, but there you don't have like sort of a ground truth estimate of the amount of signal that is there. So you don't know how much is actually there to say that you're capturing all of it. Okay, so what can you do? Well, you can say, take state-of-the-art methods and how much information can they extract from them, and then how do we do relative to those? And you're right that we don't extract as much as state-of-the-art methods, where their baseline assumption is that the molecules are completely homogeneous and static. We've relaxed that assumption to now say, okay, they can be heterogeneous and they can be flexible. And one of the consequences of that is that we don't extract high resolution information when we give it particles that are in fact static and homogeneous. And so in that sense, yeah, there is either some defect in or some lack of representation capacity in this VAE, or there's additional structural information in the images that we're not extracting properly.
I understand. Thank you for your answer. Next question is from YouTube. How did you come up with the idea of applying a voxel-based principal component analysis to relevant subsets of particle stack in your KSGA study?
Yeah, I mean, yeah, it's sort of an idea we've been playing with a bunch for other data sets. And it seemed like a nice fit here. I mean, I think, one of the ways that, yeah, it's just sort of a traditional dimensionality reduction approach. And so it seemed like the obvious thing to try, where we thought about the dimensionality of, some sort of like X by Y by Z region of space. We want to know like, what are the dominant modes of motion there? And so an eigenvolume coming out of that through PCA made the most sense to us. Yep.
Thank you for your answer. Next question is, what are the limitations of using the traditional representation of protein structure as electron scattering insancities in a 3D lattice, which is impractical for modeling multiple structures?
Traditional representation. I'm not sure I totally understand, but I think one way, I'll just try to say something that might be interesting for people to hear. If you mean by traditional representation of protein structure, might be the sort of atomic coordinates.
Yes, I think so.
Yeah. So one way, one thing you could think about is, can you come up with ways to infer the atomic coordinates from the 2D projection images directly? So the way that we frame this and the way that sort of structural biology has historically framed this is to say, you have primary data, whether it's X-ray diffraction spots or, in our case, 2D projection images from Cryo-EM. You use that data to then derive a density map. So either cryo-EM density or electron scattering potential. And then you take that density map and you use it to build a model that best explains that density map, and that model gives you atomic coordinates. And so there's sort of this firewall between the density map and the atomic coordinates. And the value of that is that, you know, if there are things that are unexpected, so say, for example, there is a post-translational modification of a side chain that you wouldn't have predicted from the genomic sequence, well, you could see that in your density map and be like, oh, look, there's some crazy, interesting PTM that occurred there, and therefore I'm going to build an atomic model for it. The alternative, which I think is what you're suggesting, is you could say, well, it's really sort of hard to generate a density map and then build a model to it. What if I just try to learn the atomic coordinates directly from the density maps? And we have another preprint where we tried to do this. I would say it doesn't work super well in that case, but you can imagine ways, you know, smarter ways to do that, maybe using like sort of biases or priors from things like AlphaFold models to get you in the right regime where you could then directly infer the atomic coordinates. And I think that there is like, it's an interesting direction of research to think about. I think the downside there is that, you know, you do sort of limit the scope of discovery you can make, because you're coming in with this strong bias of, okay, this is the linear sequence of amino acids. These are the amino acids I expect to see. And you don't have any notion of like, these are ligands, these are PTMs. Oh, there might be like translation artifacts that give rise to incorporation of the wrong amino acid at position. And maybe you want to be able to discover that because it's interesting in your biology. If your model to directly infer atomic coordinates insists that the protein sequence is the protein sequence, you're never going to see those sorts of things. So I do think that there's some value of having this sort of firewall where you first get density maps and then you interpret those density maps in the context of an atomic model.
I understand. Thank you for your answer. The next question is, why are single particle reconstruction and cryo-EM challenging for smaller sized proteins?
Yeah, so there's, it's a great question. Yeah, so the fundamental signal that we get is the difference in contrast between the protein that we're interested in and the scattering of the vitreous ice that's around it. And so as the proteins get smaller and smaller and smaller, their scattering potential decreases relative to that of vitreous ice. And so there's just less signal there to deal with. And the ways that that manifests is that it makes it harder to pick the particles, but also probably more importantly, it makes it harder to properly align them to one another. And if they're not properly aligned to one another, you don't have these poses, and then all the things that we do downstream just don't work out.
Thank you. The other question is, what makes cryo-EM the preferred imaging technique for the structural virology studies?
Yeah, so actually, yeah, it's an interesting question. So cryo-EM very early on was adopted by virologists, and I think that they recognized a couple features of cryo-EM distinct from X-ray crystallography that are beneficial in their systems. One is that you need very, very little sample, right? So for X-ray crystallography, you're typically purifying lots and lots of protein and then screening across lots and lots and lots of conditions until you find some random buffer condition that's suitable for crystallization, and then you can go and get high-resolution structures. For cryo-EM, you really just need like a couple microliters of sample, and you know, sometimes there's some optimization and getting the grids to freeze properly and stuff, but largely like you can image things pretty easily. And then for viruses, specifically symmetric viruses, cryo-EM, at least early on, the ability to get to high resolution was strongly reliant on high order symmetry, and that allows us to sort of use a big structure to pick the particles. You can get the alignments sort of coarsely correct, and then you can use the symmetry to really get the alignments proper if the molecule really is symmetric. And so a lot of these viruses are like icosahedral viruses. They're highly symmetric. And so you have a lot of subunits to align on. And so you could get the resolution relatively high even in the early days of cryo-EM on those types of particles. And yeah, I feel like there's one more thing I was going to say, but I'll leave it at that.
I understand. The last two questions. For an unsupervised heterogeneous reconstruction, how would CryoSpark and CryoDragon compare in terms of runtime?
Yeah, so CryoSpark is an incredible piece of software. It is very fast and well engineered. And so So if you compare sort of 3D variability analysis, which I'd say is sort of the most akin to CryoDragon, it's much, much faster than our approach. And so I think, even in my lab, we sort of use both. The only sort of like twist I'll put on that a little bit is that, for most of the studies that we do, it could easily be like a year of like making the strains that you need and figuring out how to purify them and getting the imaging right and doing all these sorts of things, that actually the data processing, if it takes a day to train a CryoDragon model versus like a couple hours or an hour for 3D variability analysis, it's such a small delta in the overall project time that we don't see it as like a huge defect, but it is one of the critiques that comes back often, is that 3D VA and some of the related approaches are much faster. That is true.
Yeah, I understand. The last question is, what would you recommend to students who want to study in your research area?
Yeah, I mean, I think, let's see. I mean, I think having like sort of a broad training is really useful, and being able to think about problems from different perspectives. My group is sort of a mix of experimentalists, some sort of geneticists, biochemists, structural biologists who collect data, and then also computer scientists. And so what's important is that we're all able to communicate and sort of speak some common language, recognizing that everyone has their sort of own niche where they are domain experts. But you really got to be able to talk to each other. And so part of learning to talk to each other is having a little bit broader training. And so I encourage folks early on to, like think about taking a bunch of different types of classes. If you're a computer science major, think about taking some biology classes so that you can better understand the types of problems you might want to work on. And if you're a biologist, I think it sort of behooves all biologists to think more about algorithmic design and talk to computer science folks, because the tools are so powerful and can make such a big impact in biology.
Thank you for your answer. And on behalf of myself and all our participants, I would like to thank you for spending your valuable time transferring your knowledge to us, and for this very informative and joyful presentation.
Yep. Thank you guys so much. I really enjoyed it, and great questions from everyone.
Thank you too. I wish you healthy days.
You too. Bye-bye now.
Bye-bye.